stir fry orange duck with roast potatoes and a glass of orange. Let's see what we got on here. Let's turn Facebook off now because it's 9 o'clock. See what's going on YouTube land. Caesar Milan and an aggressive pit bull. Right. You are not having my dinner. Fuck off, Scoot. Right, watch the doggies on the telly. Wait for the dog to react.
Sydney, how are you? You hold on to that girl.
My name is Sydney Namato. I am the...
Is you protecting your bitches, are you, from that aggressive male barking dog, are you? Was it a pit bull bark, was it? Get down then. Get down and calm down. Stop defending your bitches. Calm down. Calm down or you'll start the girls off. I said! She's just disciplined you, now calm down. Stop being anxious. There's no real dog. No. It was on the telly, stupid. Willow, relax, lay down. Are you waiting for the doggy to come back, are you, Willow? Are you? Ah. Oh, right, you lay down. Stop being anxious, Marnie. It was an aggressive male pit bull, I think. Alright, stop protecting you bitches. Get off my rib. Get off my fucking rib. Ah. Get the fuck off me. Ow. Get out in the hallway then. I don't mind you snuggling up against me, just not elbow in the fucking rib. Not at the moment. Alright, little lady, come on. Lay down then. Lay down.
Nani. She is my little angel, and she's only 1 year old.
Oh, it's a she.
Nani and I bonded very instantly.
I better turn it down because they're obviously reacting to it, aren't they?
Trouble started with Nani 2 months into it. We brought her to my grandparents' house and
Bouncy and energetic, isn't it?
we're all sitting in a living room, and my mom picked up my grandma's
Not
Shih
the
Tzu,
woman.
and
Like,
Nani came over
I've just
and
noticed the
she
camera angle.
started going after it.
No, I'm talking about the dog.
Nani's in the corner
Bouncing.
going after this dog and picks it up and throws it in the air. And it was
She's
just
got it on a harness first, that's wrong.
My brother grabbed my grandma's dog, and my dad pulled Nani
She's not
off,
leading
threw
from
her
the
in
neck.
pool. It was crazy. Since then, her behavior is getting
She's
worse.
letting it pull away.
Shut
up!
Even thinking about taking Nani out on a walk is very stressful.
It's a female bark,
Nani has
not a
become my
male.
boyfriend's dog 3 times.
Stop being Mr. Dominant.
The first fight that she got in with Blitz, they didn't sniff each other, they didn't do anything. She just looked at him, she looked at me, she looked at the dog again, and just attacked him. It was
He's hearing.
the scariest thing ever.
Anxious female.
The biggest fear of if Nani keeps on acting this way is that my dad will take control and put it in his hands.
Yes.
Whether it's just
It's an
driving
anxious
down
female
and
making you
deciding
anxious.
or
Now stop
opening
it.
the front door and saying, you know, she's not my dog. If that happened, me and my dad's relationship would end. I would definitely cut my life off from his life.
Get out! Get out! Get out! Get out!
So she's fighting this new energy.
Out now! Lay out.
for this new energy. But she doesn't get this energy.
Out! No, stay out. That's what you do with that anxious energy.
Control that completely, but really the real test and the real evaluation is going to be with bliss.
Go away until you calm down.
This desire of fighting with bliss.
When you get your energy right, then you can rejoin the pack. Or you start the pack off. See, Mr. Anxious, set to attention, getting anxious.
Hey. Nanny acted out. She responded to my correction. And I was able to control the aggression to a certain level. But what she really needs is a strong and consistent leadership.
Lola's watching.
What I'm doing right now is assessing how bad this thing is. It's pretty bad. Yeah, right?
Oh, yeah.
He's relaxed.
She goes from 0 to 60 in half a second. Yeah. All dogs can get there, but what makes it more manageable is it takes 3 seconds, 4 seconds, 5 seconds. And this one is half a second. Yeah.
But you can read a dog. If you can read a dog, you can stop it happening before it happens. Fuck off. Just because I've got duck fat on me freaking fingers, go away. Go away, I'm trying to watch a video. Lay down. Good boy. You can lay down there, look. I didn't say go over there, did I? I just said lay down. Want to lay between the bitches? You out. Out until I tell you can come back in. Alright, you go out there with your brother if you want to be anxious. Go and lay down. That's it. There you go, they're both being anxious now. Cheers, Marnie.
Guys, I hope you enjoyed the video. Make sure you like, subscribe, and
Because Marnie was reading the situation wrong.
Better humans, better planet.
Better humans, better planet. Because you're not allowed to be called a dog whisperer anymore. And falling out between him and the TV company or something. And they've taken the name off of him. And refused to pay him apparently. Or something like that. From what I've learnt from Caesar Malone. Bit naughty. I think that's the second part of the video that I was watching. How many days ago? 5 hours ago, yeah it is then. This is part 2 of the one that I was watching.
Viral origins, talking about the SARS coronavirus 2 virus. Where the heck did it come from?
Ukraine.
And you rumbled this.
I've seen the DOD leaked documents
Just tell us what your
saying Hunter Biden
in that
laptop.
field of study and why you were able to get to the right conclusion, or what I perceive now as the right conclusion, so much quicker than, as far as I know,
Before the outbreak occurred, they was getting funding from the DOD.
Research on HIV, and I've been working on HIV for a long time. And I had
HIV, SARS, coronavirus.
Using,
Notice why he's talking about it. Yeah, anyway, Hunter Biden's laptop.
This is the vaccine for AIDS.
For HIV.
DOD release from the US
it had 3,000 antigens
stating Hunter Biden applied for funding for a organisation in Ukraine to study coronavirus 19.
The more that we looked and analysed,
Right, which wasn't even called SARS coronavirus 19 at the time, because it hadn't occurred yet. So why would they want that in Ukraine while pointing at China?
On the white cell that the AIDS virus binds into.
Got it absolutely, and just next to it is a region called C5. And we realised that this was probably
So a virus
the Achilles heel
made
of the
from the
virus.
AIDS virus,
And we did a lot of work.
basically.
I mean, this
Didn't
was
that
over
sound a bit
months
odd?
and years and years. This was no overnight, and it was by pursuing a lot of avenues and finding it was.
That's a biological
it
weapon,
was a bit
isn't it?
like Occam's razor, that having eliminated absolutely everything else for aging, this was the only thing that stood the test of time. And it was the only thing that you didn't make responses to if you did the vaccine, because the other things drowned it all out. So I realised this was the Achilles heel. And I predicted all the failures of the virus, and I was furious because we had all our grant applications turned down by the NIH, which I now realise was Fauci. Fauci didn't understand it. The Gates Foundation, we now know the two of them have worked very closely about how things should be funded. I don't believe that if these things were properly peer reviewed, as we're told they were, that they would have been rejected given how logically we laid it out. And because of this, and all the work I'd done on this, I was contacted through an old PhD student who was working.
So I'm just eating my dinner while I'm listening to this.
Which was
Got to share the
working
info.
on an HIV vaccine with
There's
the
that
same
word again.
philosophy. Get rid of as much as you can. Go for those conserved elements that if they no longer exist, the virus no longer exists. Get rid of anything that might be a decoy. So we started collaborating. We got very good grants from the Norwegian Research Council together for doing this. The vaccine was very good. It worked very well, but it wasn't as good as we liked, but nothing is first time round. And I got involved in adapting it to make it even better. And one of the things was to add the Achilles heel to it. Their HIV vaccine was just based on the core of the nucleus, because that's very conserved. So I've added my bit in, and it works really beautifully. We've given it a booster with the IMM 101, so it's really perfect now. And it was going to go and be taken all the way up for registration by a company called Celgene, with whom I've worked with them for over 20 years. When I said, you know, thalidomide is a drug which is magical, which has been tarred by the birth defect, and you know, went through that.
This guy speaking is one of the UK's best
pay for the whole thing to go
oncologists.
through. So we had an HIV vaccine, but unfortunately Celgene were taken over by
who
Bristol
designs
Myers
and
Squibb,
makes
which is an
vaccines
enormous pharmaceutical company
to help cancer
who just
patients.
wanted their cancer agents.
He makes
They
them
took them over and
himself.
threw every other project out, which was a tragedy. However, the bottom line of that was that we were working hard together, collaborating, making this vaccine. He'd already put into the clinic before I met him, and I realised it was the right way to go. I was suspicious of the technology at first. I always am, and you always should be. Otherwise, if you take everything for granted, you end up in the mess we are now. One way of putting it. So we worked really hard on this, and we knew exactly how to do it, and he'd worked with people who had tremendous algorithms, mathematical, for picking out sequences that were really important. And he was far above, a spot above, all the so-called molecular biologists who just looked at
Did you hear that?
sequences. Because he was a biophysicist,
Algorithms
he realised that
that pick out
sequences mean
important
nothing until you
sequences.
translate them into amino acids. And amino acids mean nothing until you've built them like a Lego block into a 3D protein. And here they have their own laws. And that was the secret, that's where he was great at. So you could take the sequences and make it even stronger by making sure their charge was correct. All these things, which when I talked to other people, they didn't might as well talked over their heads. They weren't in this league. And so when the COVID sequence came along, the Børge Sørensen, who was my big friend and colleague over there, he called me up and he said, Gus, there's going to be a real need for a vaccine. Shall we collaborate and go for one along the same lines? And I said, yeah, absolutely, because I said, before we go any further, this virus has a big spike protein, rather like the HIV has an envelope. And we know the HIV envelope, using it all was a disaster. I would predict using the whole spike will be a disaster, so we need to pick the bits that are really important. So he said, great, I'll get working on those now with the algorithms and what have you. So one of the first things he did in his team was they did blast analysis, which means you take the sequence and all the proteins it could possibly make, and you look at the sequences, and the blast analysis showed that 79% of the spike protein had human homology. That meant if you use that 79% of the spike protein, there was going to be a very high chance you were going to induce autoimmune reactions against human epitopes.
Can you just unpick a minute what you mean by human homology?
Well, it means that the bit of the virus would have a very similar sequence as a very important protein in the human. And we picked out the two that had the strongest homology. One was platelet factor 4, which means the sequence is near identified. So by inducing antibodies against this, you would develop a clotting pathway. And serious, that it would explain the VIIT, the thrombocytopenic condition. I mean, absolutely explaining. This has now been accepted by the authorities as a side effect of the vaccine, albeit very rare and worth it in the long run type of approach. The other was myelin. Now, myelin is, you get that attacked, you get transverse myelitis, Guillain-Barré syndrome, etc.
So myelin is this insulating material around
The nerve.
virtually all nerve fibres.
Yeah, and that again is exactly what we see, and it's not a brand new thing, because the
Interesting.
1976 flu epidemic in America, Fort Detrick one, then that was killing young people. They rushed out a vaccine for it, and they were bright doctors, and they weren't bullied, and they reported back. Guillain-Barré syndrome seems to be much more common since we rolled out these 42 million vaccines. And they looked at this, they sent people out to check it, and they found that it was much more common. And they looked around and they said, but no one's dying of the flu anymore, it's burnt itself out. So they killed the vaccine program. That was Guillain-Barré it was being caused. It wasn't a COVID vaccine, it was a flu vaccine. So there's a generic thing between the viruses and cross-reactivity with this. Now we spotted that, so one of the questions I'd like to ask is, why did the 150 other people designing vaccines all go with the whole spike protein? And we shouted and we said, don't do it, don't do it. And because all the lemmings had all decided it was a good idea to jump with the spike protein, we were just ignored, and off they go. And every vaccine candidate uses the whole spike protein one way or the other. And we had the experience and everything to do that. But we never had the funding properly, because the people like the Faucis and Gates had refused to fund us.
So if 79% of the spike protein is the same as human proteins, and you make antibodies to that spike protein, that means to me that 79% of those antibodies are going to cross-react with human
Yes.
proteins.
But some of them will be weak and inconsequential. Some of them will be medium and cause all sorts of funny things. You wonder what's going on, like skin reactions and things which have been very common. The whole range of weird and wonderful reactions would be due to that. But the one where the homology is very strong, you're going to induce these conditions. And
This
one
is
of the
very
things
nice,
is
this
that
dinner.
always trump it. Oh, but these side effects are very
Better
rare,
with roast
and in the
potatoes
end,
than
they're worth
rice.
it. Well, if you tell that somebody's paralysed, that they didn't get COVID again because it's worth it. But to me, that was just ridiculously unacceptable when we were warned about it, and they haven't stopped the programme, knowing that the instance, it's on the MHRA website about Guillain-Barré. I mean, so that is not acceptable in young people who are, and they're often the ones that get it the worst, because they got the strongest immune response to something, who've got zero chance of dying of any of these variants. All these variants coming along, there's no evidence at all that they're ever going to be worse than the initial virus, which is why you know, we're here to talk, because it was supremely adapted to infecting and destroying human cells right from the beginning, because it had spent years being manipulated in the laboratory in order to do so.
So it's
Of
destroying
course,
human cells, the other
I've
ones.
always said Guillain-Barré, but
Sure, you're right. My French was always appalling.
no, well, my French is non-existent, so yours is clearly better than mine. But it can be a devastating disease. I've looked after patients for months on intensive care with hypoventilation, paralysis, and again, transverse myelitis. Transverse means it goes across the spinal cord. And again, I've seen people with total paralysis below that level of the lesion, just like in a spinal cord injury. These can be quite devastating diseases. Now, one of the things I think that you noticed early on was what you call viral inserts. What did you mean by viral inserts?
Anyway, back to COVID.
Right, well, this goes back to my friend Bertha Sorensen, who we're selecting, having decided that 80% or 79% of the spike protein should be avoided at all costs.
Yeah.
While putting
Isn't
that
interesting listening to two
into
doctors?
what
One-
amino acids are going to be programmed by these
One a treatment,
So these are the
Nurse come
sequences
doctor.
in the letters of the
one a
RNA
top
of the virus.
oncologist,
Yes. And
breaking it down
We
into
found
everyday language for
when you did that into the
people
amino
like me and you
acids,
to understand
there
better.
were regions where you had amino acids which had a positive charge, say 4 of them together.
Google's
Now,
not
this
going
never
to tell you the
happens
truth if you
because
look it
of
up
there's
or ask
equilibrium
Google.
goes in. There's a higher level of editing, so you have 4 amino acids positive. The editing will show that you're going to have 2 positive, 2 negative.
Yeah.
Perhaps 3 to 1 is the most extreme, not 4.
Not full.
And it was Berger who, I mean, I think he was a genius of being involved in vaccine design as a biophysicist. He said there are amino acid runs here which shouldn't be here. And I discussed them with my colleagues, and they said, oh no, it's all random mutations, it doesn't mean anything. But then we looked that they were all around the receptor binding site, and they were all heavily positively charged. Now, I'm no great biophysicist, but I could see what this meant very quickly. And we looked at the positive charge of the membrane compared to any other coronavirus or similar virus. It is unbelievably high.
So this receptor binding domain is the very bit of the spike protein that fits into
Now,
the human receptors.
my analogy, the way I saw it, and I think it's a good way to explain it, is the cell membrane for a virus is rather like the fridge door. So if you hypercharged it up, it's like a magnet. It goes bang and it locks in. That is why it's so supremely adapted to infecting human cells via the ACE receptor. In fact, it's so good, it's even learned to use other receptors, and that was the other thing that came through from that. So Berger spotted it with his team who analysed all the sequences. He spotted that these were positive charged. I went in, having spent, I discovered the receptor in 1984. So even though I was a clinical oncologist, I kept my interest in HIV all along, and couldn't understand why so many top-class scientists went down so many rabbit holes for years on end. I found that the most alarming insight that nobody was as clever as I thought they were, when I started this. But I knew the receptor.
Do you remember the
that
SARS outbreak
was involved in
in
discovering
Canada?
the CD4. Well, the first insights I had was it's not enough. There's something else required.
Way before
Something
the
else required.
coronavirus,
And we predicted
COVID-19
very accurately that there was
came
something
out as a
else
word.
required, but it wasn't discovered for
because we
another
are
10
talking
years.
about the
This is
SARS
the HIV transmission we're
coronavirus,
talking about.
Well, after I was wandering
After
around
all.
wards being a cancer doctor.
which
Yeah,
is a
being a
form
proper
of
doctor, yeah.
HIV.
And so
AIDS.
because of all that insight, I was
Same
I
family
was
of
very
germ.
well primed to understand Berg, what he was doing.
How
and
about
realised he was completely right.
Now, if
And
the
all
government
people who
told people
shouted
they
from
was
the
catching
sidelines
AIDS,
saying, "This is all nonsense and rubbish."
all in unison,
Anyway, I went and
they
looked at
fucking
papers,
panicked
I
like
read
shit, wouldn't they?
papers, And there was a
That's
very
basically
important
what
paper
happened.
came out about this time, and it was called On the Proximal Evidence. It was by a fellow called Christian
An
Anderson,
AIDS flu.
who wrote this paper with 5 or 6
How the
other top
fuck did they get it
virologists.
from a fucking
And
bloodborne
it said there's no chance
thingy
it could have
to an
escaped
airborne
from that. That was
pathogen?
their constant thing.
without
And
altering
I
it?
thought that was, after what I'd done with Berger, I thought it was absolutely rubbish. So you have
And
Which
if you
is germ
don't
warfare,
agree,
it's a
you're
biological
conspiracy
weapon, isn't it?
theorist.
if you don't agree, you're a conspiracy theorist. So anyway,
To attack
I
the
read this
autoimmune
paper,
system.
and unlike, I would say, the
Just
vast
in time
majority
for
of
a
people,
third. Well,
I actually
alright.
read their references. You always plough a load of references on the paper superficially as a decoy to make other people think you know what
Lovely
you're talking
duck
about.
stir-fry, this stuff.
That you've read everything, but in actual practice, most of the people who put their names to these things have not read them. So I took it upon myself to read them. And it was beyond belief that they quoted these papers as references, because 2 or 3 of them were really core, but they were references to bat viruses which had been genetically engineered in Wuhan labs for a gain of function, or to translate, to make them more infectious to human cells. And they described perfectly how they'd done it. They had also shown that they'd encouraged them to get even better at this by passaging them. That means putting them through all different types of human cells so they take and they adapt and they get more
So this
infectious.
is
They also, virologists,
Bill Gates and
are rather amusing.
Fauci's
They call this
organisation
polishing.
saying take it
So
further, take
you-
it further.
You polish the virus so it becomes even more.
While they
adaptable
got
to
the
cell
original lab
distancing.
And I wish
in
they wouldn't. I
Ukraine
wish they wouldn't bother.
because they
So
want
this is
them
like a
not to get
natural-
caught.
Evolution, but accelerated.
It's accelerating the evolution dramatically. So
Right,
when I
gonna
looked
hide
at these
him.
papers, I thought this insert
Smoke and
they
mirrors
just put
shit,
in
innit? Look
I thought
over there,
published
not over there.
from a Wuhan lab, it's exactly the same one that's in SARS-2. You know, and yet they're using this as evidence it didn't come from the lab.
So the same insert that was the genetic instruction, the letters, the RNA letters in the Wuhan publication were the same as the ones that turned up in the virus.
Yeah.
And strangely, I believe the Wuhan database went offline. I believe it was September 2019.
Yes, it did, and that's a very important date, the September, because just from my personal view and
It
interest,
is, yeah,
in
when you've
getting
seen the US
I was able to
DOD
identify
leaked documents
2 families who clearly
saying
went
about
down
Hunter
COVID in
Biden
November. You know, they were
getting
told they didn't
research
come in
funds
until
for
December,
Ukraine
January.
for the same
Interesting.
virus which
Clearly
wasn't
had
even
this,
a name
and,
yet.
They had all the classic symptoms. It was absolute classic. And putting my
Right.
little detective hat on, I thought,
Let's see if he
well,
comes up with
Mark,
this
you
as well.
see, What do they have in common?
That's only
What they both had in common.
That's only a
was
recent
visitors
development.
from Hong Kong. And they got ill
Interesting.
after these people came in from Hong Kong. And Hong Kong and Wuhan, you know, they're closely connected.
Yeah.
Way out London and Birmingham here, you're always going to have people back and forth. So
Interesting.
Well,
November 2019 in the UK.
here then, it was here then.
Yeah.
And the thing about the importance of September was they were very suspicious about something going on, and that's why they took it down. It was also interesting that the bat viruses that they were working on, which had clearly been harvested from a mine 1,000 miles away. It's
And
mine.
it's sort of North Thailand area, wasn't it?
Yes, and they brought them back and they sequenced, did all sorts of things that they started working on. They didn't register them in the databases until all this stuff started. Now, that's extremely unusual to do this very late registration of viruses that have been there and you've been working with. That not only made us very suspicious, but also other people suspicious who started to wonder what's going on. And the whole details of these sequences and the evolution history has actually been very well documented by Matt Ridley and his colleague Lee Shan in their book Viral. They go into that part of the evidence extremely well, which I believe is very, very complementary to the way we did it. And then after with the paper I mentioned about this Kirsten Anderson one, the one that we then found that Fauci had commissioned, they were a bit suspicious about being so black and white about it. But once he had that paper published in Nature Medicine, which was submitted and published in 5 days, something
Like I
which
said, the
never
new
happened
revelation
in a
that's come out from the
peer-reviewed
US
journal.
DOD
And
about
as soon as
Ukraine,
it was out, Fauci was waving it
Hunter
around
Biden and
Congress,
funding
NIH.
for
Here's the
COVID-19
proof. And this became embedded. Everybody was told
before
proof. And anybody who
COVID-19
said anything else was a
started.
conspiracy theory.
Well, it's got to be true because it's in nature.
And it's very true, isn't it? So I pointed out with my colleagues
Hasn't
that
been
this
made
paper is the
available
most
to this
flawed.
guy,
In fact, I
obviously,
actually said it's the worst
because he's
scientific
still
paper
going
I've
by
ever
the
come across in
original
my life.
story.
Because normally you have to search hard to find out that the references don't support the ludicrous statements, but this
And
was
he is a researcher,
glaringly
so if he
that
goes and
even
looks
I
again,
picked it
he's
up
going to
quickly,
find that
that's not-
same information,
And
which would change
they
his mind again,
turned it
and
down
you're
and
like,
said,
what the
we've got
fuck?
nothing against
Hang
your
on, we
science,
thought
but
it
it's
was
not
that,
in the
but it
public
wasn't, it was
interest
over
for us
there
to
instead.
record this.
That's what nature told you.
That's what they told me. We did the same to Science, Journal of Virology, Lancet. They all said exactly the same
It's all
thing. And
distractions.
sometimes we were rejected within 5 hours because you can do electronic submission.
Well, it's good to know that the journals have got the public interest at heart, Professor, isn't it? This is just unbelievable censorship.
The public interest, they don't want the truth out there is the only conclusion I can come to. And so if they're prepared to do that,
Why not?
with
Why
something
don't they
as
want the truth
obvious as
out
the
there?
origin
Because it's
virus, what are they
admitting
prepared to
more
do
crimes.
with everything else?
They go to jail or get hung for that.
implications, the non-pharmaceutical interventions, drug treatments, vaccines, they basically all colluded to lie to us.
Yep.
That's my take. You know, we can say I'm confused. Or
Well,
I've come to that conclusion.
You don't sound confused to me, and if we can't rely on the medical journals, I mean, you know, as someone who's, I mean, I taught evidence-based practice since it first came out in the 1990s with David Sackett teaching, and, you know, to take away this fundamental axiom of evidence-based medicine, which is the best available evidence, then we stand on fresh air. We stand on nothing.
Yeah, but they even took away the ability to debate it.
Yeah.
Now, science is all about debate.
Yeah.
It's not about censoring, and that's why. I mean, I've recently sent a book with colleagues to the press called "The Death of Science."
Yeah.
And it
Looking
was
forward to that.
because I started writing about the tyranny and nonsense, the logicality of lockdown, and a friend
Oh,
saw me doing it. And I said, "What do you think? How should we do it?" He said, "I've got a lot to add." The next thing we knew, anyway, we were editors. We were not authors anymore.
I can't wait to get a copy. When's this out?
It's going to come out very shortly. It's in the printers now, probably in November.
And if you'd like to come on and talk about it, we would be delighted to hear about it.
And it was because, you know, when I was writing about lockdown, I said the evidence is all here, but they're taking no notice of it. This really is the death of science. And my friend said, "That's a brilliant title. We're going to use this, and I'm going to expand it with you." So
Well,
that's
brilliant's one word. I would say tragic or devastating is another word. Now, there doesn't seem to be any naturally occurring SARS-CoV-2 that's ever been found in animal reservoirs or human reservoirs.
that was
Or before the
absolutely
pandemic, was there any immunological evidence of
Only
the-
simple
of such a
basic food,
virus?
but
Do you
it
think
really-
that is relevant?
really.
Well, you touched on something. It's not exactly what you asked me, but it actually helps explain very, very well what's going on here. When we had MERS, the other cattle, and we had the
This
first...
is 2001, 2003.
And then we had the SARS one.
Oh yeah, sorry, yeah.
But anyway, one had camel and the other was back to civet and things.
Yeah, the MERS was camels and the
Yes, and the
SARS
SARS
was
was
civet.
civet. Now, when you had those viruses in the tests, you could go back to the area they were found in, and you could do serological analysis of everybody in the hospital or their doctor had a blood test, and you could show there were traces there for months in the community, while said virus jumped back and forth from back to civet to human, or the camel, what's it, and it was doing...
Just got to get all the
it was
duck out my teeth now.
To get infected, to build up a viral load, whereas for the first time the disease become evident. Now that you can find for months.
Like having a second helping
In
of duck.
SARS-2, they couldn't find a single serological imprint around Wuhan or anywhere. They could find absolutely nothing, which meant it had not escaped naturally. It had gone out there full bore, able to infect and cause high viral loads in the first people that went out and hit.
And the other thing is, in previous outbreaks, we've been able to find multiple outbreaks at multiple times. So if the virus becomes endemic in a human or an animal population, then the animals could spread out over 10 miles, 100 miles, several hundreds of miles. There'd be zoonotic transfer here, then there'd be zoonotic transfer in Carlisle, and zoonotic transfer in London, and zoonotic transfer in John O'Groats, and we would get different outbreaks at different times. We didn't see that. We saw a single outbreak. Is that significant as well?
Well, I think it's highly significant. There may well have been 2 or 3 seedings in different populations with slightly different takes, but they all at the same
I call
time,
it
I believe they all
seedings.
came from the same laboratory specimen, and they found some of the first people to get infected were people actually involved in mixing the soups of these viruses. And the other thing
Seeding
I couldn't
would
understand
imply
was when we
like
pointed out
cloud
just to be
seeding.
unhappy, was the fact, no, these labs are too good. They
You get
can't
what I'm
possibly
saying
escape.
now? I'm pointing out.
When we could find 6 reports in the standard literature, 6 detailed reports of
Because
the
these
SARS
highly
virus having
educated
escaped
doctors
from
use
laboratories, one in
language
Taiwan, one in Singapore, one from
absolutely
China.
This is the
correctly.
original SARS 2003.
And this
'Cause
was
they have
meant
to.
to be highly contained laboratories. And the other thing that
They're not
was
as
very
slippery
clear
as
from a previous inspections of Wuhan was
lawyers
the
turned
level
politicians
of security
in wording and
was very,
invading and
very
concealing.
poor for a laboratory working there. So it meant that escape was inevitable in my book. So why should they cause such a fuss about it? Couldn't possibly escape. And anything to say, well, I'm a great believer in I call it the first Dalglish law of universal human incompetence.
I subscribe to that one as well.
Human incompetence is far more likely than any sort of malign intent. It may be behind it, but usually what you end up dealing with is a piece of human incompetence.
Yeah.
Which was the same with the foot and mouth outbreak too.
Yeah. So the 3 options for the viral origin are natural zoonotic, accidental lab leak, or deliberate lab leak. And I think I'm with you, accidental lab leak.
So that's
Notice the wry smiles
that's where I
from
came
both
from,
of
that this
them.
was
Accidental lab
a
leak.
lab leak, and the problem was they tried to cover it up.
Yeah.
Instead
Yeah.
of coming clean. And here is something really, really.
You've got to watch what you
interesting.
say, haven't you?
They actually got the WHO.
You see what I mean?
into going along with them trying to cover it up.
Oh, my
If the
vape's charged up.
WHO had been under the previous aegis of the previous head, whose name just escapes me, who was not under the control of China, like Tedros was, and who had never been head of WHO. He wasn't even a medic. He was put in there for political reasons. He did what he was told. Shut up, didn't do it. Had they done their job, this would never have escaped to the rest of the world. I'm talking to lots of people involved in these things. I completely believe that. So
It
I
would have
think
been contained in Wuhan area,
in Wuhan,
Hubei area.
yeah, and it would never have got outside China.
Stop the flights from China. And the WHO at the time specifically said we should carry on flying in and out of China. Even at that time, I was bemused.
That was madness. Just clear, obvious madness. But the real sinister thing here is that the WHO said in order to handle this better next time, we need all the powers assigned to us.
Right.
That is, I would rather sign a suicide pact
Yep.
with something. UK.
Well, never let a good crisis go to waste, Professor.
Well, hopefully they've got to name
See
it
what I
here,
mean about the wry
come to
smile?
our senses.
Same one as when they was answering
I
that
don't
thingy,
hold
wasn't it?
out much hope. The people who seem to runners
So I was
advising
correct.
things seem to be the most inappropriate people, and the government seems to be so poorly educated,
Ironic
so
answer.
intellectually lazy. They go along with all this.
Yeah. And we have done several videos on that. People like James Roguski who's done absolutely sterling work on that. Now, I keep getting questions on this. I've never fully understood it. How similar, how homologous are SARS coronavirus 2 sequences with the HI virus,
With the which virus?
the human immunodeficiency virus?
Oh, sorry, yeah, the HIV virus. Sorry,
Yeah. I got told off once. A virologist told me off. He says, I said the HIV virus. He said, how can you have the HIV virus? so since then, I've always tried to say HI virus. But,
Right. I've always just say HIV.
yeah.
There is very, very little homology, but there
Okay.
is a tiny little bit of homology. And this bit of homology is real, but I think we've discovered why it's there. When I mentioned about how they get their bat viruses and they train them to infect human receptors, they put the human ACE receptor into a cell, a neutral cell, with an HIV lentivirus. So the virus takes the receptor in, it gets integrated up, and then it gets expressed on this cell that would never normally present it. Those little HIV sequences it was an HIV lentivirus. So when you look at the sequences, you're picking up the sequences from the vector the ACE receptor was introduced to the cell with. The virus has picked them up. So the homology is really, there's nothing to do, there's no link with the two. But those sequences, to me, absolutely confirm that it was not only genetically engineered, but it was heavily passaged through that. And that's because of that passaging, it's picked up those HIV sequences. That's purely all it is. There's no big homology with the two.
Right, good. Now, my last question is, I've only ever partly understood this. There's a furin cleavage site on the spike protein of the virus. And furin is a protein on the surface of human cells.
Mm-hmm.
Now, this furin cleavage site, I kind of think of it as kicking open these furin doors to allow the virus into the cell so it can reproduce. So is that kind of vaguely right, and what is the significance of finding the furin cleavage site on the spike protein?
The significance of the furin cleavage site on spike protein of the SARS and MERS virus group is it's never ever been near any of these viruses whatsoever, ever.
So just to clarify that, sorry, the MERS and the SARS coronavirus 1 don't have a furin cleavage site?
No, neither do any within that phylogeny of the coronaviruses from which these viruses have been.
So SARS coronavirus 2 is the only coronavirus that we know of with a furin cleavage site?
Of the sort that can infect man.
Yes.
There are some animal coronaviruses, which my virology friends keep proudly telling me do have furin cleavage sites, but they don't infect us. This is the only one that does. Now, the furin cleavage site was put into these bat cells by people in the
still.
Wuhan lab because they boasted about it, that they did it. These same people, when they presented the sequence of the virus in nature, the furin cleavage site is screaming in plain sight. They discuss everything else about the sequence, but they don't discuss the furin cleavage site.
They probably forgot.
Oh, you're very kind. They forgot. They put it in there.
You couldn't make this up, could you?
No. And what a furin cleavage site does is basically if you have a virus getting onto the cell, you can lock on pretty tight, but you have to work pretty hard to get through the cell membrane. But a furin cleavage site is like giving the virus an extra pair of scissors, so it can just slip through and get into that very, very quickly. That never occurs in this type of virus. It was put there in the Wuhan lab, and it was they who it's like magic.
What's that look for? You ready to come back in here now, are you?
Can
you stop being silly then? Come on then.
Pointed it out.
Come on.
And then we found that other people have pointed it out. David Baltimore was a famous virologist. He said it's very odd. He called it the smoking gun. And this went as far as the Washington Post and this, that, and it was all dismissed as nonsense of no importance. And then
Get
another load of virologists come up and said, well, some viruses have these, which is true. But
Get
not the sort of viruses
off.
we're dealing with infecting
Get
humans. It's completely
off.
irrelevant.
It's like saying
Well,
some
you want to
animals have
smell me
trunks,
breath?
and humans don't.
Oh, okay. Yeah, we have to do this thing every time we eat some food. We have to blow the breath at the dog.
To go against our,
So they can
our
check what I'm
discussions
eating.
here, which other people joined.
They like to be
slowly,
nosy.
They said, oh, but we found this virus is present in
Right,
the
you're going to
raccoon.
sit down there,
So
are you,
this
Hulkster?
was
Right,
the
come on
case
then,
for the
lay down.
raccoon.
Give me
So
some elbow room.
they look around,
Move.
all the raccoons know this,
Lay
this
down.
is the only one. It could be a false positive.
Sit. Stop getting
And you get
fucking antsy.
papers coming. There was a paper from,
Good boy.
say, a city in Scotland which tried desperately hard to open up the narrative that it was a natural jump, what have you. And by this time, I know how to do my science. I just looked at their funding. China. It's ridiculous. They don't even have to go to the referee.
First scientific analysis is these days. Who the heck paid for this?
Who is funding? And unfortunately, it's even more important when you get into treatments and vaccines.
They didn't eat their dinner till late yesterday, the boys.
I don't want to get into that now, but the
seem to
reason
be
I'm so
preoccupied
concerned
with the
about the
girls at the
viral
moment.
origins is
Rather
it
than their
possible that
food.
humans could be
Unless I
jiggly
put meat
pokering
in there.
around with a virus which is as
Oh, you're going to
transmissible
fidget
as,
and
say,
mess about, are you,
measles
Hulkster?
or an Omicron
Okay, you're going to
virus,
lay
but
between
as
the
deadly
doors,
as,
alright?
say, an Ebola virus.
You want to
such
lay
a thing
like
possible?
that, okay.
Yes, it is.
Good
This
boy.
is all called the gain of function. And this is what, under the Obama
Gain of
administration, they
function.
banned. And the scientists, and I've got a colleague I know myself.
Gain of
basically,
function.
He calls all virology gain of function in a desperate attempt to try and preserve it. No, it is, I mean, the sort of thing that was being done.
You do want to
absolutely
preserve
mad.
your seed,
It
don't
was
you,
basically-
Hulkster?
It was inevitable that a pandemic would arise out of this nonsense. Obama people banned it, but they tried to get an exception, and who was the person who tried to get the exception? And that was Anthony Fauci, in order to have things in case something dreadful came along. Very exceptional cases. Well, he wasn't allowed to do it in his own place, so he funded this work to be done in Wuhan via the Eco Health Alliance, via a person called Peter Daszak, who is very, very friendly with Batwoman, who is the woman whose lab did this very manipulation that led to it coming out, to escaping. So when Fauci went to these extreme lengths to deny this, I immediately knew he was covering his backside over this. But he realised he was in trouble. So whereas initially I blamed the Chinese, for which I was called by some people a racist, I don't know how, when you actually point out that something comes from somewhere, you're a racist for pointing it out. But when I trace everything back, if somebody has to take the rap for this, it has to be Fauci. To me, he is the one single person guilty. He's just used his excessive, outrageous power, an unbelievable war chest, to bribe and get what he wants. And I use bribe succinctly and clearly because Kirsten Anderson, who wrote that ludicrous paper for him that he got the narrative, immediately got a nearly $9 million grant to carry on his work in his institute, which did not go out to peer review in all the normal circumstances according to my sources. And,
Well, just
about
another
that.
bizarre temporal correlation, isn't it?
and the same happened with Daszak. The same happened with Daszak. They wrote a letter to the Lancet saying this is absolutely ludicrous. Anything that suggests it came out of China lab is, and they used the word, it was racist and didn't mean it. God, everything else.